Hello and welcome to my channel. In this lecture we'll continue our discussion of blood elements, so let's begin. The blood cell, also called hemocyte or hematopoietic cell, it's produced in the process of hematopoiesis in the bone marrow. And the major types are red blood corpuscles and white blood cells and platelets. And together the blood cells constitute 45% of the blood tissue by volume. The remaining 55% is being plasma, the liquid form of, or the liquid component of blood. The red blood cell, it's better to call it red blood corpuscle because it lacks the nucleus and the organelles. Um, um, and it's you can consider it a sack of hemoglobin that is rich in iron and capable of binding oxygen to transport it to the different tissues of the body. The cell membrane of the red blood corpuscle is composed of proteins and lipids, and the structure of the membrane provides the durability or deformability and stability of the necessary for the blood corpuscle to survive, especially during a passage into capillary network, because during capillaries they squeeze themselves to pass there and return their original shape after passing. The red blood corpuscle is flexible, oval biconcave disc. As mentioned before, it lacks nucleus and most organelles to allow for space for the hemoglobin for the maximum capacity of oxygen carriage, and the lifespan of the RBC averages 120 days before it's destroyed by macrophages. The red blood cells constitute nearly half of the blood volume, and the packed red blood cells you will encounter in hospitals are red cells that have been processed and stored in blood banks. They are called packed RBCs. The membrane composition, the membrane is responsible for the blood corpuscle to deform itself and the flexibility of the blood corpuscle, and also responsible for adherence to the other cells, and also able to interface with immune cells. It's composed of three layers: an outer glycocalyx, which is mainly carbohydrates, a middle bilipid layer, and with transmembrane proteins, and the internal skeleton is a network of protein, and it's on the inner surface of the bilipid layer. So it has three layers: glycocalyx, bilipid layer, and inner surface proteins. The membrane proteins, as mentioned before, responsible for deformability and flexibility, durability of red blood cells, especially during passage in capillary networks, because the red blood corpuscle has to squeeze itself in a capillary that's around half its diameter and retain its discoid shape after passing the capillary network. The proteins present in the membrane, there are around 50 known membrane proteins. 25 of these membrane proteins carry the blood grouping antigens, such as the antigens for A and B and the Rh antigens. The membrane proteins can perform other functions such as transporting ions and molecules, adhesion and interaction with other cells like endothelial cells, and receptors also. And the blood types of humans, the variety comes from the variations in the glycoproteins present on the membrane of the red blood cell. White blood cells, on the other hand, are called leukocytes. They comprise the immune system, they're important for defending the body against infectious diseases and foreign invaders. And they all have nuclei, different than the red blood corpuscles, of course, and platelets. You can classify the white blood cells according to their cell lineage into myeloid cells and lymphoid cells, and according to structure or granulation into granulocytes and agranulocytes. The myeloid cells include neutrophils, eosinophils, mast cells, basophils, and monocytes. You can subdivide the monocytes into dendritic cells and macrophages. Dendritic cells, this is a picture of dendritic cells. These are cells present in tissues in contact with external environment, like the Langerhans cells in the epidermis, which you can see abundant Langerhans cells here in this section of epidermis. And this is a picture of a macrophage. The lymphoid cells, on the other hand, they include T cells, which can be divided into helper T cells, memory T cells, and cytotoxic T cells, and B cells, divided into plasma cells and memory B cells. And in addition to T cells, there are the natural killer cells. One of the ways to classify white BCs according to granulation into granulocytes and agranulocytes. The white blood count, the white blood cell count, is essential to know the status of a disease in a patient, for example. The normal count being 4 to the times 10 to the power of 9 for each liter, and they make approximately 1% of the total blood volume, of course lesser than the RBCs. Increasing the number of the leukocytes, it's called leukocytosis, and it may be normal as a healthy immune response, or abnormal in neoplastic and autoimmune diseases. On the other hand, decrease of leukocytes below the limit is called leukopenia, and this means that there is a weak immune system. Types of white BCs, they are all nucleated, and according to the structure, they are divided into or classified into granulocytes and agranulocytes. Granulocytes can be distinguished from agranulocytes by their nucleus shape. Their nucleus is multilobed, or called polymorph polymorph nucleus. It's different than the agranulocytes. And by also cytoplasm granules, presence of cytoplasmic granules all over the cell. So the granulocytes are polymorph nucleus, have polymorph nucleus and granuloses all over the cell. In the myeloid cell lymphoid cell classification, myeloid cells are neutrophils, monocytes, eosinophils, and basophils, distinguished from lymphoid cells by the hematopoietic lineage, by studying their lineage. Lymphocytes can further be classified as T cells and B cells and natural killer cells. And this is a picture showing the different types of the white BCs. Now, the first type is neutrophils. They are the most abundant among the cells. They defend the body against bacterial infection or fungal infection and form pus after phagocytosing the organism because they die. They die after phagocytosing few pathogens because they cannot renew their lysosomes like monocytes, for example. So they have short lifespans, but they are the major component of pus. And they are referred to as polymorph nuclear leukocytes. They have multi-lobed nucleus that looks like multiple nuclei, but it's a single nucleus divided into lobes connected by strands. And the cytoplasm may look transparent because of the fine granulation that are pale when stained. Eosinophils, on the other hand, they fluctuate, the levels of them fluctuate, seasonal fluctuation throughout the menstruation and throughout the day. And they are concerned with allergies and parasitic infections, collagen diseases, diseases of the spleen and central nervous system. They are more numerous in the mucous membranes of the respiratory and digestive systems and the lower urinary tracts. The function primarily deals with parasitic infections, and also they are inflammatory cells, important inflammatory cells in allergic reactions. They destroy the large parasites like the hookworms and tapeworms, and the nucleus is bilobed, connected by a thin strand. The cytoplasm is full of granules that have a characteristic pink-orange color with eosine staining. This is a picture of eosinophil. Basophil, responsible also for allergic and antigen response, and they secrete histamine and heparin as part of their function. The histamine acts as a vasodilator of the nearby capillaries and blood vessels, leading to passage or more permeability to cells necessary to defend the body against infection. And also the heparin is an anticoagulant that prevents clotting, blood clotting, and promotes movement of white blood cells into the area. They are rare type of white blood cells and can be recognized with their several dark violet granules that the granules almost hide the nucleus, which is bilobed or trilobed. Basophils also can release chemical signals that attract eosinophils and neutrophils to an infection site. This is a picture of a basophil. Now, lymphocyte, they have a deep staining nucleus and relatively small amount of cytoplasm. They are divided into B cells and T cells. This is a picture of B cell here. The B cell binds to pathogens and activates complement system in the immune response and ensures pathogen destruction. While the T cells, on the other hand, can be divided into helper T cells, cytotoxic T cells, gamma and delta T cells. The CD4 helper T cells, these are cells that express the receptor CD4, and this receptor is able to bind antigenic peptides presented on the major histocompatibility complex class 2 cells. Class 2 cells that responds to agents from outside the cell. Helper T cells also make cytokines and perform other functions that help coordinate the immune response. The T cells are called cytotoxic. These are the CD8 positive. They have the co-receptor CD8 interacting with major histocompatibility type class 1, which is present on virus infected or tumor cells. And major histocompatibility, we'll talk about them in the next slide. This class 1 presents antigen from inside the cell, not from outside the cell. The gamma and delta T cells, alternative T cell receptors other than the CD4 and CD8, and it's found in tissues more commonly than in blood. This is a picture of a T cell, and this is another picture of an RBC, a platelet, and a T cell. Now, I thought it's better to give a hint about the major histocompatibility, class 1 and 2. There are molecules on top of cells, a cell surface of all nucleated cells in the body. This is major histocompatibility class 1. The class 1 is made of 4 domains, 3 alpha domains and 1 beta domain. And the site for binding the peptide is between alpha 1 and alpha 2, as you can see here. Their function is to display peptide fragments of proteins from within the cell to cytotoxic T cells. That is CD8 T cells. This will trigger immediate response for the immune system. CD8 T cells, as mentioned before, interacts with major histocompatibility class 1. While class 2 molecules found only on professional antigen presenting cells like dendritic cells and mononuclear phagocytes and thymic epithelial cells and beta and B cells. And the cells, of course, is a component of the immune response. And the antigens presented here is from the extracellular proteins, not cytosolic as in MHC class 1. It's made up of 4 domains, 2 alpha and 2 beta, and a peptide binding groove between alpha 1 and beta 1, as you can see here. Natural killer cells are able to kill cells of the body that do not display MHC class 1 molecules. Decreased expression of MHC class 1 happens when the cells are infected by a virus or a cancer. The monocytes are the largest type of white BCs. They are phagocytic functions like the neutrophils, but have longer lifespan because they can replace their lysosomal contents. They have the kidney-shaped nucleus and typically are agranulated. They eventually leave the bloodstream and become tissue macrophages. They remove the dead cell debris as well as attack microorganisms. They present pieces of pathogens to T cells. This is a picture of a monocyte. The last type is fixed leukocytes. These are leukocytes that migrated into tissues and permanently resided there. We have an example as the von Kupffer cells in the liver, for example. They have a role in the immune system, like the histiocytes. The histiocyte is a tissue macrophage. Dendritic cells, we talked about dendritic cells before. Mast cells, cells that connective in the cell in the connective tissue that contains many granules rich in histamine and heparin. Microglia, they are present in the brain and spinal cord. For dendritic cells, we showed you the example of Langerhans cells in the skin, for example. Langerhans skin, for example, and they are present also in the nose and lungs, mucous membrane, and stomach and the intestine. I hope you got the maximum benefit from this lecture, and thank you very much for watching.